Good morning. Welcome to the session number 10. Today I'll be continuing with the same chapter, that is chapter number 6, that is molecular basis of inheritance. Today I'll be discussing lac operon concept. Lac means lactose, operon means it is a group of structural genes. It was proposed by Jacob and Monod. Jacob and Monod proposed a model to study the regulation of gene action in one of the bacteria that is said to be Escherichia coli. So it is the regulation of gene action in this bacteria that is said to be Escherichia coli. Now coming to the structure of this E. coli. There are three divisions. Here also three. This is regulator and this is promoter and this is operator. Regulator, promoter, operator. And this is lac Z, lac Y, lac A. This is the Escherichia coli. It consists of two types of genes. These three are said to be structural genes. Structural genes, and these three are said to be control gene. Control genes. So, lac operon consists of two types of genes, control genes, structural genes. Control genes, there are three types: regulator, promoter, operator. Together is said to be control genes. And the structural genes, there are three types: lac Z, lac Y and lac A. Now coming to the first one. This lac Z that mainly helps in the synthesis of an enzyme that is said to be beta galactosidase. This enzyme mainly helps in the breakdown of lactose into glucose and galactose. So this is the first type of structural genes, lac Z, that helps in the synthesis of beta galactosidase, so that it mainly helps in the breakdown of lactose into glucose and galactose. Coming to the second one, that is lac Y. This helps in the synthesis of permease. This enzyme mainly helps in uptake of lactose from the surrounding media. In the media there is a presence of lactose, that in that media lactose is there, so that this enzyme permease that helps in uptake of this lactose from the media. Coming to the last one, lac A. This helps in the synthesis of trans-acetylase. This function is not known. So now these three together that mainly helps in the synthesis of mRNA. So that the structural gene, already I told you, operon means it is a group of structural genes that mainly helps in the synthesis of mRNA, there are three types of gene that is associated here. The first is the LAC-Z, helps in the synthesis of beta-galactosidase, so that it helps in the metabolism or breakdown of this lactose into glucose and galactose. And the second one is the LAC-Y, helps in the synthesis of permease, so that it helps in uptake of lactose from the media. LAC-A, this helps in the synthesis of trans-acetylase, so the function is not known. Now, these three structural genes together helps in the synthesis of mRNA. Now coming to the LAC-operon concept, how it works. Sometimes the genes become passive and sometimes the genes become active. That is said to be what you call regulation. So, LAC-operon concept consists of control genes, structural genes. Now, here, first is in the absence of inducer. That means lactose. In the absence of lactose, what happens? This regulator is denoted by I, promoter is by P, operator is by O. In control genes, there are three types: regulator, promoter, operator. Coming to the first is the regulator. Regulator, it helps in the synthesis of a protein that is said to be repressor protein. Promoter, where there is a attachment of RNA polymerase enzyme, always binds to this part. Operator act as a single switch for the synthesis of mRNA. These three are the main functions. I repeat, regulator helps in the synthesis of repressor protein. Promoter, there is a presence of an enzyme RNA polymerase that gets bind to this part. And last is the operator, it acts as a single switch for the synthesis of mRNA. It acts as a main switch for the synthesis of mRNA. Now, this regulator helps in the synthesis of repressor mRNA, which in turn produces a protein that is said to be repressor protein. Now, this repressor protein binds to the operator. Here, there will be a attachment of this repressor protein so that the operator turn off, so that the switch off. So that means there is no synthesis of mRNA. So what happens? Regulator that continuously synthesizing repressor mRNA, which in turn synthesizes repressor protein. As a result of this, this protein immediately binds to the operator so that the switch turned off. So that means there is no synthesis of mRNA. Now, in the presence of inducer, this is again Escherichia coli, 3 units. Here also 3. LAC-Z, LAC-Y, LAC-A. Same thing here, I, P and operator. Here what happens? In the presence of inducer, this regulator continuously synthesizing repressor protein, it immediately binds with an operator so that the switch turned off. So that means the genes become passive, that means there is no synthesis of mRNA. So in this case what happens? Here also the same thing, this produces, that means the regulator produces repressor protein. Now this binds with the operator like this, binds with the operator. Here, presence of lactose. Jacob and Monod introduced lactose in the media. It acts as an inducer. This lactose is also known as allolactose. This lactose now introduced into the media. Now this lactose binds to the repressor protein like this. When this lactose binds to the repressor protein, there will be a structural change that takes place in the repressor protein so that it immediately separates. So the switch turned on. That means there will be a synthesis of mRNA. This is how the regulation of gene action that takes place in Escherichia coli. I repeat, lac means lactose, operon means group of structural gene. Here, this is an Escherichia coli, consists of structural gene, control genes, structural gene that mainly helps in the synthesis of mRNA. There are three types of structural gene, lac Z, lac Y, lac A. These three are mainly responsible for the synthesis of mRNA. Coming to the control gene, so that it controls the structural gene. There are three types: regulator, promoter, operator. Regulator that produces repressor protein, promoter RNA polymerase enzyme attached to this part so that it helps in the synthesis of mRNA. Operator, it acts as a single switch for the synthesis of mRNA. Now, first is in the absence of inducer. Here the lactose acts as an inducer. Without lactose, what happens? Now, this regulator that helps in the synthesis of repressor mRNA, which in turn synthesizes repressor protein. Now this repressor protein, what happens? Immediately it gets attached to this part, that is operator. When this repressor protein attach or bind to the operator, the switch is turned off so that there will be no synthesis of mRNA. So in the presence of inducer, now this is the same thing, regulator, which in turn produces repressor mRNA, finally produces repressor protein. Repressor protein binds here so that the switch is turned off. In the media, Jacob and Monod introduced lactose. So this lactose now binds with the repressor protein. As a result of this structural change that takes place in the repressor protein, immediately this repressor protein separates so that the switch turned on, which indicates there will be a synthesis of mRNA. This is how the regulation of gene action that takes place in Escherichia coli, and it was proposed by Jacob and Monod, and it is said to be lac-operon concept. Lac means lactose, operon means group of structural gene. So in the presence of inducer, what happens? Lactose binds with the repressor protein so that there will be a structural change that takes place in the repressor protein so that it immediately separates from the operator so that the switch is turned on. This is about the lac-operon concept. Next, we will move on to genetic fingerprinting. Next, we will move on to genetic fingerprinting or DNA fingerprinting. by Alex J. Jeffrey. So, coming to the DNA fingerprinting, also known as genetic fingerprinting by Alex J. Jeffries. As the name itself indicates, DNA fingerprinting or genetic fingerprinting, it's the technique to identify the individuals. That is mainly based on the DNA sequences. So, what is meant by genetic fingerprinting? It is the technique to identify the individuals that is mainly based on the DNA sequence. That is said to be, what do you call? DNA fingerprinting or genetic fingerprinting. Here, human DNA consists of 6.6 into 10 to the power of 9 base pairs. Here what happens? 99.9% of this is said to be non-coding sequences. Non-coding sequences. And only 0.1% is said to be coding sequences. That means 99.9% is said to be non-coding sequences and it is common in all the individuals. This is common. Coming to the second one is 0.1% is said to be coding sequences. So that means it codes for a particular protein and this is very unique from one individual to another individual. Based on this, they are going to identify the individuals. So here 99.9% is said to be non-coding sequences common in all the individuals. And only 0.1% is said to be coding sequences that codes for a particular protein and this 0.1% always varies from one person to another person. Now, if the DNA is isolated and it is subjected to density gradient centrifugation, what happens? Here we will get two types of peak like this. This higher peak is said to be bulk DNA. Bulk DNA. And the smaller one is said to be satellite DNA. So this bulk DNA is nothing but 99.9% and this satellite DNA is nothing but only 0.1%. DNA is isolated and subjected to density gradient centrifugation. What happens? We'll get two different peaks. This is the higher one, elongated one is said to be bulk DNA, that is nothing but non-coding sequence. So that it is common in all the individuals, and very smaller peak that is said to be satellite DNA, that is nothing but the coding sequence. This is very important and this is very unique in individual to individual. So coming to the satellite DNA, satellite DNA is nothing but the DNA which contains repetitive sequences. Repetitive sequences. Satellite DNA is nothing but the DNA which contains highly repetitive sequences are said to be satellite DNA. Here there are two types of satellite DNA. One is said to be mini satellite, microsatellite. Mini satellite, microsatellite. Mini satellite, here the nitrogen bases that may varies from 10 to 60 base pairs. Here it contains only 2 to 6 base pairs. Both are nothing but which contains repetitive sequences. So the nitrogen bases here that contains 10 to 60 base pairs, 2 to 6 base pairs. And here this mini satellite that repeats may be 10 to 1500 times repeats. Here 10 to 200 times repeats. And this mini satellites commonly said to be variable number tandem repeats, and this is said to be microsatellite is said to be short tandem repeats. Variable number tandem repeats, short tandem repeats. What is meant by this variable number tandem repeats? Tandem repeats means one after another. Here the base pairs usually that contains 10 to 60 nitrogen bases. For example, here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For example, G, C, G, C, A, T, C, G, A, G. This is about the particular mini satellite, and this contains about only 10 nitrogen bases, and this unit is repeated number of times. That is said to be mini satellite, also known as variable number tandem repeats. It may repeat from 10 to 1500 times. Here short tandem repeats which consist of only 2 to 6 base pairs. For example, G, T, C, G, like that. G, T, C, G, same thing. G, T, C, G. This is about the short tandem repeats. So these tandem repeats or variable number tandem repeats always varies from person to person. Because of this variable tandem repeats, in forensic science they are going to identify the individuals. So now what is meant by satellite DNA? Satellite DNA, that is nothing but the DNA consists of repetitive or highly repetitive DNA sequences. There are two types, mini satellite, microsatellite. Mini satellite that consists of 10 to 60 base pair, here 2 to 6 base pair, here 10 to 1500 times it repeats, here only 10 to 200 times repetition. This is said to be variable number tandem repeats. This is said to be short tandem repeats. Now coming to the procedure. How they are going to identify the individual. Coming to the procedure for DNA fingerprinting or genetic fingerprinting. First is the collection of sample from the crime spot. From the crime spot, they will collect the sample either in the form of blood or in the form of any type of a small tissue, or hair, or nail, or semen. These are all nothing but sample. So the first is collection of sample from the crime spot. Then after the collection of sample, the DNA is isolated by using refrigerated centrifugation. So after refrigerated centrifugation, what happens? The DNA is isolated like this. This is the isolated DNA. So first, collection of sample from the crime spot, either in the form of blood, semen, tissue, hair, nail, etc. So here what happens? After the collection of sample, DNA is isolated by a method that is said to be, what do you call, refrigerated centrifugation. And here this is the isolated DNA. isolated DNA that contains variable number tandem repeats. Now, isolated DNA is treated with restriction endonuclease enzyme so that it recognizes the particular sequence, and this results in the formation of cutting the DNA into DNA fragments like this. This is the third step. First is the collection of sample. Second is isolation of DNA treated with REN. There will be a formation of what? DNA fragments. Now, coming to the next step, these fragments are placed on an agarose gel. This is agarose gel. Here, small wells are created. To this, we have to introduce this DNA. And this is negatively charged, and here this is positively charged anode. So now what happens? There will be a separation of DNA fragment according to their length, and this process is said to be electrophoresis. So, this DNA fragments now introduced into the agarose gel. So, there will be a separation of DNA fragments according to their length. The process is said to be electrophoresis. Now, to this, alkaline solution is added. As a result of this, what happens? The double stranded structure now becomes single stranded structure by breaking the hydrogen bonds. Now, after this electrophoresis, these DNA fragments are placed on nylon membrane. This is said to be nylon membrane, which contains artificially synthesized single-stranded radioactively labeled DNA. These are said to be props. So, what are props? Props are the single-stranded artificially synthesized DNA fragments. Radioactively labeled DNA, that is said to be props. So, here what happens? After electrophoresis, these DNA fragments are transferred to the nylon membrane, which contains artificially synthesized single-stranded radioactive labeled DNA are said to be props. So, this process is said to be southern blotting. Name of the scientist who identified this, therefore it is said to be southern blotting. So, this results in what? Hybridization takes place. That means here there will be a pairing takes place. Like this, because it always follows the base pairs. So, after southern blotting, what happens? Here, this is nothing but radioactively single-stranded DNA props, artificially synthesized, known as DNA props, and these props are immediately get attached to the normal DNA fragments. This is said to be hybridization. X-ray film is placed on the nylon sheet. What happens? There will be a presence of bands like this. We can see the bands, and these bands are nothing but DNA fingerprinting. DNA fingerprinting. And this process is said to be autophagy. radiography. So what is meant by autoradiography? After southern blotting, there will be an hybridization takes place, and then X-ray film is placed over this. What happens? We can see the very much elongated bands, and these bands are nothing but DNA fingerprinting, and the process is said to be autoradiography. Now, this band's position, length, thickness of these bands always varies from one person to another person, so that these bands are compared to the bands that are present in the suspect. If the two bands are identical, then he is the culprit. If the two bands are non-identical, he is considered as an innocent person. This is how in forensic science they are going to identify the criminals. So I'll repeat. First is the selection of sample from the crime spot, either in the form of tissue, blood, semen, hair, nail, etc. This is subjected to refrigerated centrifugation so that the DNA is isolated. DNA now treated with restriction endonuclease, and this results in the formation of smaller fragments of DNA. These smaller fragments of DNA now placed on an agarose gel so that there will be a separation of these fragments according to their length, and the process is said to be electrophoresis. And this DNA or the agarose gel is treated with an alkaline solution so that the double stranded DNA fragments now become single stranded by breaking the hydrogen bonds. Then these DNA fragments are placed on a nylon membrane which contains artificially synthesized single stranded radioactively DNA. Artificially synthesized single stranded radio labeled DNA. These are said to be props, and the process is said to be southern blotting. So after southern blotting, what happens? There will be a pairing takes place. That is nothing but the adenine always pairs with thymine, cytosine always pairs with a guanine, so that it becomes double stranded structure. And this is this nylon membrane. So what happens? The X-ray film is placed on this so that we can easily see the bands. And these bands are nothing but DNA fingerprinting, and the position of the band, thickness of the band, length of the band always varies from one individual to another individual. So these bands are compared to that of the suspect. If the two bands are identical, then he is considered as a culprit. If the two bands are non-identical, he is considered as an innocent person. So this is about the genetic fingerprinting. So I completed the chapter number 6, starting from the DNA. What is meant by DNA? What are all the components that is present in DNA? And how these components assemble together to form a nucleotide, and how this nucleotide that are arranged to form an elongated structure, that is nothing but the structure of DNA, Watson and Crick model, how the replication takes place. That one of the important method is said to be semi-conservative method of replication, and I hope you understood an experiment to prove that the replication was said to be semi-conservative, and then you studied about transduction experiment, transformation experiment, and then RNA, what are all the components of RNA, and the different types of non-genetic RNA, mRNA, messenger RNA, ribosomal RNA, and transfer RNA. These three synthesized in the nucleus, these three now enters into the cytoplasm so that it helps in the synthesis of proteins. And then coming to the next is the transcription and translation. Together is said to be central dogma. It is the movement of genetic material from the DNA into the protein, so that it includes two steps: transcription, translation. Transcription, it is a portion of the DNA that take part in transcription, that is said to be transcription unit. And then we studied transcription in prokaryotes, transcription in eukaryotes. Very, very important. And then coming to the translation, how there will be a synthesis of proteins in the cytoplasm. And then we studied genetic code, nothing but a group of 3 nitrogen bases that is mainly found on mRNA that codes for a particular protein. And then we studied diagnostic characters of genetic code. That is also very important 5-mark question. Then we studied Human Genome Project, salient features of Human Genome Project, goals of Human Genome Project, diagnostic characters of human genome, and some of the applications. And then in today's session, I covered lac operon concept in Escherichia coli, how these genes are get activated and in some cases it becomes passive, that is said to be regulation of gene action in Escherichia coli. And then coming to the last part is the DNA fingerprinting or genetic fingerprinting, that is nothing but there is an identification of a particular individual mainly due to the presence of particular DNA sequences, that is said to be DNA fingerprinting. I hope you understood this chapter number 6, that is molecular basis of inheritance. Thank you.